So in the next two lectures, what I hope to do is to first review some basic concepts about immunology, and then we'll look at how viruses interact with the host immune system. So what this is doing, what I'm hoping to do through these initial series of lectures in the course, is to give you sort of a broad overview of the steps necessary to carry out a viral infection. So we've seen already how viruses interact and enter into the host. We've seen the basic steps of viral replication within a cell, and we've seen how viruses exit from the cell. So what we need to just overlay that with is the idea of what happens to the virus when it interacts with the host immune system. And once we've done that, we've covered the general steps or layers involved in infection of a host and spread of a virus. What we'll then do in the remainder of the course is to sort of overlay this basic understanding into specific cases about particular viruses or unique viruses. But what I want to do again in this first part of the course is just to lay that groundwork for the overall understanding of the processes necessary to carry out viral replication. And so in today's lecture, we're going to focus on a basic understanding of the immune system, because really the immune system is tied to the viral replication cycle. Viruses and their hosts have been evolving together over millions of years, and so our immune system is actually a product of that battle waged between viruses and the host. So our immune system has been sculpted by viruses, and viruses have been sculpted by our immune system. And much of the concepts of virology rely on an understanding of how viruses and the host, particularly host defenses, how that interplay works. Our immune system is divided up into two broad components. There's the adaptive immune response and the innate immune response. So the innate immune system is characterized by a lack of memory, so it doesn't improve with repeated exposures to the same pathogen. The innate immune response is always on and often represents the first barrier to viral replication. So we've already talked about briefly in the last lecture about things like surface barriers. And surface barriers are a prime example of the innate immune response. Right? So the skin represents an impenetrable barrier to viral infection. Remember, skin is composed, the outer layers of skin are dead cells. Remember, viruses require living cells to carry out their replication. And so if there aren't breaks or abrasions in the skin, a virus can't penetrate the skin, can't cause an infection. So viruses either need to get through breaks, nicks, damage to the skin, or they need to find another portal of entry. And oftentimes viruses will enter through mucosal surfaces. This slide just shows you some of those barriers to infection. And so we have skin and mucosal secretions, cilia, and we talked about those in the last lecture. Okay. Those are examples of innate defenses. They don't improve upon repeated exposure to a pathogen. What I want to spend a little bit of time on, because we've sort of already touched a little bit on those surface barriers, are nonspecific cellular or chemical defenses. And these are innate immune responses. Again, they're pathogen independent, but they're mechanisms that the body has to deal with infection. Okay? And so some of these nonspecific defense mechanisms include things like producing fever, coughing, sneezing, inflammatory response. Those all represent nonspecific chemical or cellular defenses. And I want to spend a little bit of time talking about the interferons. The interferons are a unique class of proteins that are turned on very early in response to a viral infection. Now, the interferons were discovered in 1957 through a really simple and elegant experiment. So what scientists did is they took choriallantoic membranes and use those as a substrate for viruses to infect. Now, prior to the development of cell culture, viruses were often cultured on chorioallantoic membranes from chicken eggs. And as you'll see when we talk about influenza virus and the influenza virus vaccine, the vaccine is actually still produced on chicken eggs, which is a throwback to this sort of old technology of culturing viruses. So what scientists did was they took 2 test tubes and they put chorioallantoic membranes in each of those tubes. To one of the tubes they did nothing, so they mock treated it. To the other tube, they added UV inactivated viral particles. Well, what does UV inactivation do? Well, UV light damages the nucleic acid, and so although the virus can enter into the cell, it can't carry out replication. So the DNA or nucleic acid is damaged, and so it can't produce proteins and it can't replicate. But the virus itself is able to enter into the cell. So one tube was mock treated, another was treated with UV inactivated viruses, which can enter but can't produce infectious virions. Okay. After that treatment, both tubes were treated with wild-type virus. What they found was that the mock-treated tube, the virus was able to replicate, but in the tube that had been pre-treated with the UV inactivated virus, the subsequent addition of infectious virus or wild-type virus, the virus didn't replicate. So what was going on? Well, scientists ultimately purified a protein which they called interferon because it interfered with the ability of viruses to replicate. And interferons are small proteins. They're a member of the cytokine family. And cytokines are sort of the communication molecules of the immune system. It's the way various components of the immune system talk to each other. What was happening was interferons were being produced upon treatment of cells with the UV inactivated virus, and that interferon was protecting other cells from subsequent challenge by infectious virus. There are a whole series of interferons which have now been identified. The one that relates to viral infections is interferon alpha and interferon beta. And the interferons bind to the interferon receptors, and they elicit what's known as the antiviral response. So what do we know now about the interferons? Well, what interferons do is interferons are produced by a cell that's infected with a virus. Interferons are soluble proteins, and so a virally infected cell will secrete interferons into the media, and it will bind to interferon receptors on surrounding cells. Binding of interferon to the interferon receptor sets up the antiviral state in surrounding cells, such that those cells are resistant to viral infection, which is what happened in that original experiment. Pre-treating cells with UV inactivated virus caused the production of interferons, which then set up an antiviral state in all the surrounding cells, which prevented those cells from being infected by the wild-type virus when it was subsequently added. So what do interferons do? How do they set up that antiviral state? Well, there's 2 components that I'll mention today, although interferons produce a complex mixture of changes within cells. Okay. One set of changes that interferons produce is the presence of an RNase. An RNase degrades RNA. And as we talked about already, many viral genomes are based on our RNA genomes. So genetic material is RNA. So if interferon becomes activated, RNases get turned on, which degrade RNA. Now when an RNA virus tries to enter into the cell, right, it becomes degraded by its genome becomes degraded by the RNases. And so the virus essentially loses infectivity. The other thing that happens upon binding of interferon to its receptor is the shutdown of host protein synthesis. Okay. And as we've already talked about, all viruses require the host translation machinery to carry out their protein synthesis. And so interferon shut down that host protein synthesis, with the net result that when a virus tries to enter into a cell that's been stimulated with interferon, if it's an RNA virus, its RNA genome will be degraded, and when it gets into the cell, no proteins can be produced because the host translation machinery has been shut down. So essentially the cell becomes resistant to viral infection. And the idea is that one cell may die, so one cell becomes infected, but it sets up this antiviral state saying, whoa, there's a virus in the neighborhood, be prepared. And so all the surrounding cells become resistant to infection. It's a very powerful defense against viral infection. But it's really a non-specific defense. It doesn't matter what type of pathogen is present, the interferons will behave in much the same way. So what actually triggers the production of interferons? Well, there are a couple of triggers, but one is the presence of double-stranded RNA in cells. Why double-stranded RNA? Well, that's because double-stranded RNA is not normally found in a typical eukaryotic cell. So think about it. What normally happens in a cell is you have DNA transcribed to RNA translated to protein. So what you normally have in a cell is single-stranded RNA, the mRNA within the cell. So how do you get double-stranded RNA in a cell? Well, that happens primarily when a cell becomes infected with a virus. So if you imagine a single-stranded RNA virus enters into a cell, and that single-stranded RNA virus needs to replicate itself. Let's say it's a positive sense RNA virus. That positive sense strand needs to be converted to a negative sense strand, right? And then the negative sense strand is used as a template to make more positive sense strand. So to replicate a positive sense genome, you need to make an intermediate molecule, a negative sense strand. So you have a positive sense strand used as a template to make negative sense strand, which is used as a template to make more genomic copies of the positive sense RNA. Because you have both positive and negative sense RNA within the cell, it can hybridize together to form double-stranded RNA. So the only time you should have double-stranded RNA in the cell is if something is wrong. The cell is infected with a virus. And so it's a way that the cells can recognize that they've become infected with a virus. And again, it's part of the innate immune response because it really doesn't matter what virus is doing it. As long as the virus produces that signature of double-stranded RNA, interferon will be produced in the cell, and then the interferon will spread to surrounding cells to set up the antiviral state. So interferons represent one of the first responses, host responses to a viral infection, and a very powerful response against viruses.